Next one up is Gregor Fuhrmann, and he actually has a title that I always claimed the opposite. I said that extracellular vesicles are not really suited for the small molecule drugs because I think they have a role in RNA and protein, but not really small molecule drugs. But I'm happy to be convinced by you in the next 10 minutes or 15 minutes or so.
Thanks. Yeah, so I hope I can convince you. Good morning also from my side. I'm very happy to be here and to present some of our work on the extracellular vesicles. So in my group, we're interested in developing biomimetic systems, so systems that are present in nature, that use principles present in nature. We want to use them for improved diagnostics or imaging, but very specifically to improve the targeted drug delivery, to improve the tissue specificity, reduce accumulation in other tissue, reduce side effects overall, to improve the therapeutic outcome. And so we're very specifically interested in those extracellular vesicles. We've heard a lot about these. They're produced by cells. They're composed of a phospholipid bilayer membrane decorated with surface and membrane proteins. They can also have a natural cargo, and we know, or we think, that they're important for intercellular communication. What's also interesting is that we find them in almost any body fluid, blood, urine, breast milk, and so on. So they seem to be stable under these physiological conditions, which is very interesting from a drug delivery point of view. What's also interesting is that upon a certain pathological trigger, they can appear in a higher number, in a different composition, in a different size, and so forth. So this is information that can be used to develop sensitive and early diagnostic tools. We also think that they do have some advantages over these so-called classical delivery systems. They have a natural composition. They do have an inheriting targeting mechanism. We hope that they'll be less recognized by the immune system. Some of them also have some inherent mechanism to overcome biological barriers. And you can also see them as a kind of first step towards a more personalized medicine. If you use cells from a patient, isolate the vesicles from that patient, load it with a drug of choice, and then give it back to that patient, you might reduce side effects, you might reduce immune activation, and overall improve the therapeutic outcome. So we wanted to look at small molecule compounds and to see if those vesicles are useful as carriers for these, because there's a lot of new drugs that have developed, small molecule drugs. We all know that many of them suffer from low water solubility, which is a big problem in order to bring them into the cell, in order to bring them to their site of action. So we isolated vesicles from different sources, explored various loading techniques to incorporate model compounds, and then we looked at cellular uptake and also the therapeutic efficiency of those drugs. And the drugs that we chose were porphyrins of different chemical composition, but with very different hydrophobicity. So a very hydrophobic one, a hydrophilic one, and then an intermediate one. Moreover, those porphyrins are also fluorescent, so we can detect them without additional labeling once they enter the cell. And they're also explored as for photodynamic therapy. So if we shine light on them, they will produce reactive oxygen species that can then kill cells, for example, cancer cells, but this might also be useful for treating infectious bacterial infections. So we also, the vesicles, we isolate them from cell culture supernatant. We use a differential centrifugation protocol to spin them down and to isolate them from the supernatant. And we looked at different types of vesicles from cancer cells, endothelial cells, and then two types of stem cells, embryonic and mesenchymal stem cells. And we imaged their size and size distribution with this technique that I think many of you are familiar with, this nanoparticle tracking analysis. So it's a single particle tracking method that not only gives us the size distribution, but also the number of vesicles, which is a very important parameter that we need to know. And I think maybe with the exception of the embryonic stem cell vesicles, they all show a rather narrow size distribution, given that this is a natural product. We then also confirmed their shape, their morphology, and with EM imaging, we see this typical cup shape that is also reported in literature. So just to confirm that we actually do have extracellular vesicles here. We then first looked at their stability, and we saw that just storing them at 4 degrees in the fridge in PBS, for some of them they're stable up to 2 weeks, which is already pretty good. We're now extending this, looking at different storage modalities. In particular, we're interested in looking at lyophilization. It seems that some of these vesicles even have an inherent stability for lyophilization to protect or to be stable in terms of size and in terms of number. This is an ongoing work from one of my master students. So we then looked at loading of different model compounds. This is an example for the very hydrophobic porphyrin, and we saw that just by passive incubation, so just mixing the vesicles with the hydrophobic compound is enough to allow integration of the compound probably into the membrane of the vesicles. If you want to load more hydrophilic compounds, you need a kind of active loading technique to kind of open the membrane. And this is a method, electroporation, which is a short electric pulse that transiently opened the membrane so the drug could go inside. And in our hand, this worked pretty well for increasing the loading efficiency for those more hydrophilic compounds. We then also looked at cellular uptake. After purifying the vesicles by size exclusion, we threw them back onto cancer cells. And in these 2 top images, I think you cannot see very well, but they're massively taken up into those cancer cells. Much better compared to the free drug, also compared to very plain liposomes that we did not further optimize. This is basically the quantification of those images here. And you see that even if we increase the amount of free drug by 10 times, we don't get quite the same uptake of drug compared to our vesicles. So we can load them with small model compounds, and they bring their cargo, they deliver their cargo at the cellular level, which is very interesting. When we started imaging those cells and looked at kinetics of uptake just by microscopy, we noticed that just the light exposure from the microscope itself is enough to activate the drug, to activate this production of reactive oxygen species. Because what we saw is that cells started to die. This is an example here. This cell and this cell, they have the fluorescent drug. And you can see that this cell starts to not look very healthy, and then also this cell at some point will undergo apoptosis, while, for example, the control cells that are just incubated with PBS, they happily proliferate. There's no impact of the microscopy itself. So we started counting cells, counting the time of death, and we calculated that the cells that had previously been incubated with different types of drug-loaded vesicles, they do have an increased risk to undergo apoptosis compared to our control samples. So the vesicles deliver their cargo, the cargo is still active, we can still use it for therapeutic application, which is also very important. And then finally, we also wanted to look at more hydrophilic compounds and explore additional loading techniques for these. And I'm going to point out 2 examples here. This is the loading efficiency of the various methods. What's worked pretty well was a treatment of the vesicles with a mild surfactant, a saponin, but also using a hypotonic dialysis that would induce swelling of the vesicles so they take up the exterior medium. Both methods work pretty well in terms of loading efficiency, at least compared to the electroporation, also compared to the passive incubation. But then when we looked at cellular uptake, only the vesicles that were loaded with the saponin method showed a significantly better cell uptake compared to all the other methods. And we're now also extending this using this mild loading technique, using the saponin for larger biomolecules, and they are still active afterwards, and we're not the only one using this method, so it seems to be a very interesting approach to load vesicles. So to sum up, I think those vesicles are very interesting entity of nanoparticles. They seem to have a natural role in delivering information between cells. They have some interesting properties in terms of drug delivery, in terms of size, drug delivery properties, stability. We've created a tool set for loading of model compounds ranging from very hydrophobic to hydrophilic drugs so you could basically choose the drug of your choice and encapsulate it into the vesicle that you want, and then look at cellular uptake, cellular interaction. And in our case, the incorporation of those porphyrins not only increased the cellular uptake of the drug, but also improved the therapeutic outcome for a photodynamic therapeutic approach. So with this, I'd like to finish. I would like to thank the people that were involved in this, especially my master student Julia, also my small group at the Helmholtz Institute for Pharmaceutical Research in Saarland. Thanks, of course, for the financial support, and thanks for your attention. I'm very happy to answer any questions.
Wow. Well, thanks a lot. And indeed, well, what is at least convincing to me is the fact that many people in the field use passive incubation, basically, to load their vesicles. And to me, that all doesn't really make sense, because if it passively goes in, it also passively goes out. And there's hardly any opportunity for the vesicle to do its action. And the fact that it's really dependent on your loading mechanism, I think that is really convincing and helps a lot to really make this, this approach work. Questions? Dominique. Sorry. We still have to figure out who does what.
Yeah, thanks, Gregor, for this nice talk. Do you have any idea why these saponin-treated extracellular vesicles are taken more up by the cell?
At least I know, I can imagine why the other methods don't work that well. When the other method that we tried, we saw really morphological changes in the vesicles. So I think the other methods are just not as good for loading the vesicles, so they kind of lose their biological activity. And I think the saponin is just mild. It apparently does not interfere too much with the protein conformation composition on the surface, so they still have their inherent interaction abilities.
Okay, thanks.
Yeah, is it important to get them in the same size? Because I guess there is a distribution of sizes for those guys. When you apply them for delivery, you may have this heterogeneity may affect biodistribution. Can you really, for example, using extrusion or some other method, to make them all the same size?
We tried, but particularly the extrusion changes the conformation, so they look weird in the EM imaging. For the size, I don't know. I think this is the same as Ray already said at the beginning. There might be a subpopulation that works very, very well. There might be some subpopulations that don't work well. Of course, this is a bulk. It's a bulk effect, but I mean, you've also seen that the size distribution is not that broad in our example. So, yes.
Thank you. So, Gregor, what do you think, this seems like an obvious question, but does like go to like? In other words, does an exosome from a tumor cell, when it's loaded, go preferentially there, or can you use any exosome from a T cell or a dendritic cell or any other kind of cell?
Yeah, that's a very good question. So we looked at uptake of, in this example we looked at uptake of different vesicles from different sources on a few different cells. There is not a big difference in the overall amount that go into the cell. There's difference in the kinetics, so some go in very fast, some very slow. I would also think that tumor-derived vesicles, they would invade other cells much more, because I think that's also their natural or pathological role, to attack like normal healthy cells, lyse them or destroy them to make space for the tumor itself. But there is examples of very specific interaction. For example, vesicles from, I think, T cells and antigen-presenting cells, they very specifically interact. So, yes, I think it depends a lot on the source. It's not just vesicles. You have to be, specific.
My question is very different. When I hear about using extracellular vesicles for drug delivery, people focusing on technological issues, how to make enough, how to get drug into it, and so on. My question is, how do you think that it can be possible to make reproducible batches and to document what is needed for drug approval?
I think we can, I mean, this is a question that comes up quite often. I think we can take advantage of industrial scale processes that are already used, for example, for biological drugs that are recombinantly produced. There is first approaches on having really clinically great vesicles from a defined set of cells. There's approaches for immortalizing certain cells to culture them in larger quantities. So there is approaches that are currently being developed, and I think we can, I mean, for biological drugs it works as well, so I think we can take advantage of that. We can also take advantage of the preparation of liposomes. We can combine it with engineering vesicles. I think that probably a semi-synthetic way might also be an option, but this is still underway. Yes, we do.
We have a discussion planned at 3 o'clock, and I think it's outside in the foyer.
Yes.
We'll have to see how we organize that, but I think that is really one of the key issues that we have to discuss. How are we ever going to
Yes.
take the next step into clinical application?
Okay. Great talk. I have a question actually related to the source. So when you take like a source from tumor cells or from other cells, there is a lot of content like microRNA, RNA already in these cells. So when you collect from one type of source and then you put it in patients or in animals first, how these RNA and microRNA or other proteins which are being carried by these exosomes will affect the cells apart from your drug?
I don't know. So there's two things on that. What we did is we also just took the tumor cell-derived vesicles and threw them on healthy cells. We only left them there for 24 hours, but we did not see major changes in the cell viability and in the cell morphology. Of course, that would have to be extended. Also, what I'm not sure is when we treat the cells to load them, it might be that the cargo is somehow destroyed, is somehow lost, because it's a treatment, for example, with a surfactant. It might also have an impact, but that's definitely something you would have to look at in terms of safety and biocompatibility. And I don't think tumor cell-derived vesicles would be the therapeutic form at the end. I think we'll have to look for a different cell source, stem cells, for example.
My question is actually a follow-up question on that, because we know that exosomes, and you said that only a small population is finally arriving, the rest is going somewhere. We know that exosomes are involved in creating the niche of metastasis. So the problem is that let's say what I see is that we are running in the same problem like we have with viruses. That we always have, let's say, the cargo which is inside. We don't know exactly what it's doing. We know that there is most probable an effect of the sum of these vesicles. We know that some have the properties you want, but you don't know what the rest of them is doing. And in the worst case, you are creating a platform which then in the patient is creating the niche for metastasis or for cancer.
I agree. I mean, it depends a lot on the very well characterization. It depends, as Imre said, on the purification methods. But there is examples of like people isolate vesicles and they inject them into patients. They can, for example, reduce immune response. There's an example for host versus graft reaction, and it was a massive reduction. So there is examples. Of course, this is all coming up, but yes, it needs to be addressed. But I think that's the interesting part now, to really characterize very well, define isolation techniques that are standardized so that everybody does the same thing, have specific type of cell source.
The problem is the timeline. Remember how long it needs before a patient developed cancer. So the problem is if it's possible for you to see the connection between the injection of the exosomes and the extracellular vesicles and the development of cancer 10 years later. It's a long-term process, and this is, let's say we have no model systems at the moment which can image or which can mimic these conditions. That's one of the main problems. Sorry.
I think that's the perfect start for the discussion in the afternoon, I think. Yeah.
Already alluded to a bit the fact that, well, to step away from the drug delivery field, what is sure is that the extracellular vesicles have a biological effect, and it doesn't need to be efficient in order to be biologically relevant. And I think the next talk by Professor Burlingham is really one of the, I thought it was really an exciting paper to read about the extracellular vesicles mediating microchimerism and split tolerance. And well, I'm looking forward to your talk.